Hello, everyone, and welcome to part one of the lecture on chapter 3, Molecular Biology, Genetics, and Genetic Diseases. This PowerPoint was created by the authors of our text, and I have further edited it. And here we go. Some key concepts. So this is all about genetics. You have learned about genetics before in your biology studies. I'm going to remind you of a lot of terminology. DNA is the big molecule that has all the genetic information. Resides in the nucleus. It has to be well protected in there. But we need the information that is in the DNA in order to make our proteins. The sections of the DNA that actually code for proteins, that's what we call genes. And they're responsible for our ability to put amino acids together and make proteins. Sometimes the genes get messed up. There's a typo that happens when it's being copied or it's damaged by radiation or chemicals. Those changes can be permanent. We call those mutations, and they can change the primary sequence. What that means is the sequence of amino acids. And why that matters is these sequences of amino acids determine the shape of the overall protein. And if that mutation happens in just the right spot where the protein's supposed to be shaped like this and now it's shaped like that, then that protein can't do its job anymore. That's a real problem, and that is a genetic disease. I'll remind you, we as humans get half of our genetic information from our moms and half from the dads. Those chromosomes combine to form a zygote. And when we try to follow through the family tree information about who has what medical condition or what genetic disease, this is what we refer to as a pedigree. And it's sometimes the first step that healthcare providers can take to try to start figuring out what's going on. And then, of course, there are other laboratory-based methods for detecting genetic disorders. Some of the big disorders, I'm not going to read them all. You can pause the recording and read them yourself, but we're going to hit on some of these big ones, and we will mention some others. This information is covered on these pages. Oh, that's the general pattern. So I will have page numbers on the bottom that are usually highlighted in yellow. Those are the pages in our textbook where you can read some more about this information. All right, cellular information transfer. This one's a little bit tricky. I'm using an image from a freshman level biology text just because I think it's beautiful and clearly illustrates things. But this material is covered in our textbook on pages 39 and 40. So here's a cartoon that shows generally the cell cycle. The cells spend most of their time in what's known as interphase, and there's for a long time, people thought nothing was really happening here. They said there was a gap in the activity. That's what the G stands for. And then looking through a microscope, they saw that there's a whole lot of other stuff happening at this part of the cell cycle, and they didn't quite know what was happening. But then things seemed to slow down, and G2, that refers to the second gap in the activity. Well, it turns out that there's a whole lot going on during G1 and G2. We just didn't know what it was. But what's happening at these two extremes of the cell cycle are during S phase, that is when the DNA is being copied. So all that material, all that information that's in the nucleus, we need to make a full copy of that before the cell can divide into two cells and grow, the body can grow. And so we see the DNA gets copied at S phase, and at the opposite end of the cell cycle, that's when mitosis and cytokinesis is happening. That's when the chromosomes are lining up down the middle, and then they pull away from each other. You remember that from 100 years ago, mitosis and cytokinesis. Okay. So as I said, the DNA has to be copied. So you start with the double-stranded DNA, but then you make a second double-stranded molecule, and they're still stuck together. When you go from DNA and you copy it and make more DNA, that step is referred to as replication, or DNA replication, or DNA synthesis. And then this is the more famous part of things. DNA is used to make a kind of copy, but it's not exactly that same copy, to make a molecule called RNA, that molecule is allowed to leave the nucleus, which it has to do if this information is ever going to be used to make a protein. This process, taking information from DNA all the way to making a protein, that's referred to as gene expression. You're taking the code of DNA and using it to make the actual protein. So the gene is being manifested, if you want to say, made something physical. You're expressing the information that's inside the DNA. So when do these things happen? Oh, sorry, names. All right, I'm sure you learned these names before. Transcription and translation, people get them mixed up like crazy. The easiest way to remember is, nobody forgets this part. You go from DNA to RNA, that's easy. And then the next step, you go to finish the process, that's to make the protein. But you'll get the terms mixed up. If you do, the easiest way to remember which one's which is they are in alphabetical order. Transcription comes before translation. So that's how you can remember now, forever. So when do these things happen? The timing of this, making the copies of the DNA to make more DNA, that only happens during S phase. A common misunderstanding is that people think that it happens during mitosis. No, you have to have that DNA all copied. You have to have 2 full sets of information before the cell is allowed to start going into even thinking about making 2 separate cells. Okay, this other thing, this gene expression, that is happening all the time during G1, during G2. It's always ongoing. All right, so the cellular transfer of information, as we've said, the DNA can copy itself during S phase, but if you're hoping to be able to make a protein, that information has to leave the nucleus. So the DNA has to make a version, a different oh my gosh, it's a slightly different molecule that is actually allowed out of the nucleus. That is the RNA. That's the transcription step. And then that RNA, in order to finally finish this process of gene expression to get to be made into a protein, that has to connect with organelles called ribosomes. Let's see, the message that's carried in this RNA, that's referred to as messenger RNA. There are a couple of other kinds of RNA that we will touch on. Covered on page 38. DNA structure, backing up a little bit. This is a super stable molecule, the double helix. It's held together by these hydrogen bonds. I think you probably remember letters A, T, G, and C. The A's and the T's connect to each other. They make 2 hydrogen bonds together. We say it's complementary bonding. And the C's and the G's go together. Those have 3 hydrogen bonds between them. And I kid you not, that is how the DNA can copy itself, because when you pull those 2 strands apart and you're going to go make new strands on this one, this side has a T. Well, if I'm going to build a complementary strand, I know I need to bring an A in. And on the other side, where I have the A, to make a complementary strand, I need to bring a T in. And those will connect because of the 2 hydrogen bonds that are going to form. Easiest way to remember, if you have to, is the pointy letters go together, A and T are pointy, and the curvy letters go together, C and G, they're curvy, they go together. All right, size, billions of base pairs. So those A's and T's, that's a base pair. The larger structure we call our chromosomes. There are 46 of them in most typical humans. 46, that means we have 23 pairs. 22 of those pairs are the non-sex chromosomes, and the 1 pair of sex chromosomes, either XX, meaning a biological female, or XY, typically a biological male. The other 22 chromosomes are referred to as autosomes. They're numbered from 1 to 22 just based on how big they are. We all have the same biggest pair that's referred to as chromosome 1. And that's just by convention. We all agree to call them that. It's not like they come in order or anything like that. Of those 2 copies of each chromosome, we get 1 from dad and 1 from mom. The structure, the DNA, how it gets coiled up to make these really tight chromosomes, it's wrapped around these proteins called histones. And historically, we didn't think that they did anything. We thought that was just like the toilet paper, like the cardboard inside the toilet paper roll. We thought that was it. Turns out the histones are super active in determining which genes are expressed at what times. So we're learning more and more about the way gene expression works, and a lot of it has to do with what these histone proteins are doing. Normally, when the chromosome is in the nucleus, all protected in there, the chromosome is loose. It's not tightly bound. And because it's loose, it is available for transcription to start that protein production process. The gene expression is what I'm trying to say, where you go from the DNA to make the RNA, and then the RNA can leave the nucleus and go be used to used as instructions for making a protein. Well, you can read more about it on these pages. Okay, so the cell cycle. I showed you one cartoon. This is the cartoon our textbook uses. We have the cytokinesis. The cell starts in G1. This was the gap in the activity. Then the DNA has to get copied. That's the DNA replication during the DNA synthesis phase. Then this other gap in activity, it looks like nothing's happening. And then finally we get to mitosis, and that's when the cell divides. It turns out this is all super regulated. So cells are not allowed to move through the cell cycle without meeting these very strict conditions. So for example, the G1 and S checkpoint, the cell does not even let itself start putting energy into copying DNA. That takes energy. That takes molecules of material. Like, this is ecologically expensive job to do. And so if the cell has no chance of being able to reproduce and actually separate into 2 cells, that cell will not initiate DNA replication, will not copy the DNA. This is how people end up with stunted growth. If they don't end up getting sufficient nutrition at the right age, they might not reach the full potential of how tall they could have possibly gotten. And it's because of this checkpoint. Because if there's insufficient nutrients of any kind, the cell says, nope, there's no chance that we're going to be able to make 2 cells, and just making more DNA is a giant waste of time and energy. So we're not doing it. That's fantastic. Some more things that are shown here. This is not explained in the book right now, but I'll mention it in passing. So there are some cells, we made all of our cells. At one point, all of us were just 1 cell big, and look at us, we're lots and lots of cells. But there are some of our cells that cannot make copies of themselves anymore. They're stuck in this gap in activity, their G1 phase. It's referred to as G0 or G0, that the cell there's no way that it can replicate the DNA, and it can't undergo mitosis, can't divide. What cells are those, you're thinking? Haha. Think about the places in the body that can get damaged and cannot be repaired. At least right now, we don't know how to make them fix themselves. If you are thinking cardiac muscle cells and neurons, you are correct. That, my friends, is why heart attacks are so dangerous and why strokes are so dangerous, because the cells are here in the cell cycle. They cannot move forward into DNA synthesis and then moving it along. Fascinating. Yeah. yeah, there's plenty of research into this area, because clearly at some point the neurons could copy themselves and the cardiac muscle cells could copy themselves, because like I said, we started out as one cell, and look at us with these giant brains and these big hearts. Somehow the cells are able to copy themselves, but when we reach a certain age, the cells seem to be permanently in this position. I'm putting this bug in your ear in case you know any researchers. These are still open questions. We're interested in figuring out, how can we knock a cell out of G0 and back through the cell cycle so that we can make more cardiac muscle cells, we can make more neurons, so that the spinal cord injury isn't as devastating as it is right now. Okay, enough about that. DNA replication, okay, yeah, energetically costly. And there's some other checkpoints. So here's another one. If the chromosomes are not aligned properly, then the cell, or sorry, if we don't have two copies of the chromosomes, then the cell does not move from G2 into down this way into mitosis. And then there's another checkpoint here during mitosis. If the chromosomes are not lined up correctly, if the spindle fibers, if they haven't grabbed a hold of the chromosome to pull them apart, that cell is not allowed to go through mitosis. We will get back to this when we talk about neoplasia, when we talk about cancers. All right, different kinds of cell division, mitosis. This is for growth and repair. This is where we start with a cell that has two copies of every chromosome. That's 46 chromosomes. And when they have two copies each, this is referred to as diploid, Di meaning two. And when that cell divides, it's magic. You still have two copies of every chromosome. And how this magic happened was during the synthesis phase, each chromosome doubled up, but it was still attached. And then during mitosis, the two homologous, or the two chromatids, they separate from each other. And so each daughter cell still has a full complement of information. So that's how all of our skin cells have 46 chromosomes, and they give rise to more cells with 46 chromosomes. In contrast, there's another kind of cell division. It is only performed in the ovaries and in the testes, and it's only for making eggs and sperm. So this kind of cell division is meiosis. That generates a cell that has half the normal number of chromosomes. So from 46 down to 23. That's the term for that is haploid. And what is that good for? Well, when you have a 23 chromosome cell get with another 23 chromosome cell during fertilization, then you're back to the diploid number. So that's how the zygote ends up with 46 chromosomes, which is the normal number of chromosomes. We can go into the details of meiosis 1 and meiosis 2. We're not going to go into that kind of detail, but I do want you to understand that it's very important in the formation of the egg and the sperm that the chromosome number decrease down from 46 down to 23. Accidents can happen here. And when accidents happen here, we end up with an egg or a sperm that has the wrong number of chromosomes in it. And that has devastating consequences for the zygote that is formed. All right. Finally, recombination. This is how chromosomes, two of the same chromosome, like we said, the biggest chromosome, we call that chromosome 1. So when you get the chromosome 1 that came from mom and the chromosome 2, chromosome 1 that came from mom and chromosome 1 that came from dad, and they get together, they actually can do this trick where they swap pieces of each other. It's a weird little thing that happens, but they reconnect and they'll swap ends. And so you end up with what's known as recombination. And why that matters is this is how when a mom and a dad get together and they have a kid, and the kid can look like all 4 of the grandparents. It's because of this kind of recombination. It leads to more genetic variability. Okay, let's see. We talked about how the whole point of having DNA is so that you can use that information for making proteins. But we can't go directly from DNA to protein. We have to go from DNA to transcribe it to make RNA. This is the messenger RNA. And then the end of that gene, there is something called a stop codon that says, okay, stop copying this gene here. This is the end of this. And that's how the RNA polymerase, you don't do like transcription of the entire chromosome. You just do it snippets of the chromosome. That's what we call a gene. Okay, and then the RNA, messenger RNA is processed a little bit. It has to leave the nucleus, goes out to the cytoplasm to begin that next step, that translation step. And that is what happens with the ribosomes. So with the help of this ribosome, represented by this giant pink box here, it clamps around the messenger RNA, and here's where the magic happens. Remember, we said we're going to go from DNA and RNA to proteins. And the big difference here is all you see on the right side of the screen were lots of little letters. This is the messenger RNA. Then what we're making using this information is this chain of amino acids. That's what's going to become our protein. And so this is where the magic happens. We have something called a transfer RNA or tRNA. Its 3-letter code here has to match the 3-letter code on the other side. And why that matters is because it's holding on to a particular amino acid. I'll remind you of some more biochemistry. Amino acids, there's 20 of them. They're shaped slightly differently, and it's based on that shape that you end up with a protein that folds and turns and holds a particular orientation and can do a particular job. Like that was supposed to be an enzyme, a digestive enzyme chopping on something. Okay. And so anyway, this is how that translation step happens. Here's the interpreter, if you will, this transfer RNA. It reads the RNA on one side and it brings in the correct amino acid on the other side. All right, so different transfer RNAs, they're built differently and they hold on to a particular amino acid. Like I said, there's 20 of them. Here's the table. I'm not going to make you memorize this, but I do want to point out a couple things. One is, so the words, if you will, they're 3-letter words, and they're referred to as codons. If the first letter of the word is U, and the second letter of the word is C, then we go over here. And then the third letter of the word can be any of these things, and it still means I want the amino acid serine. So there's some overlap, there's some sloppiness, if you will, in this code, where you can pronounce it this way, or you can pronounce it this way, or you can pronounce it this way. It all means serine. Bring me the amino acid serine. Okay. However, if you get to, I don't know, trying to think of C2 that are similar to, oh, okay, here's one. All right, so if instead of UCU serine, I have UAU, that means bring me this other amino acid. Oh, actually that, when there's a typo, I mentioned that there could be typos in here. If we change that middle letter from a C to an A, holy smokes, that means we changed from the amino acid serine to the amino acid tyrosine. Depending on the shapes of these amino acids, that could have huge consequences for the shape of the protein. Here's an even bigger mistake. If you have, let's see, UAC, it's supposed to bring the amino acid tyrosine, but instead I goofed and I made a mistake. And instead of bringing the amino acid tyrosine and then continuing on with the rest of this gene, instead it says, no, instead of UAC, it says UAA. That doesn't mean bring me an amino acid. That means stop right here. I don't care that this the rest of that strand is there, and there's all this information in there. If we got a stop codon kind of mutation, that means stop right there. So you're like part of the way through making this protein. Well, you're out of luck, because that particular mutation is devastating, because now it's not going to make the protein at all. Okay. Why do I make a big deal about all of this codon, this giant table? Because your amino acids were built, put together in a sequence to make this big protein based on this table. And whether it's your gene or my gene, we're using the same table. It's not just that. Your genes are your codons and my codons, and the codons of the cow also same codons. Your codons and my codons and the codons of the bacteria, same codons. It's the same exchange. If this sequence says UUA, that brings a leucine, and it's going to bring a leucine whether it's in me or it's in you or it's in a cow or in a bacterium. Wait, there's more. This is how viruses get us to make their proteins for them. We all use the same table. We all use the same translation system. And so this is how a virus can inject its DNA or can inject its RNA into us. And then we go ahead and make the proteins for the virus. That is amazing. And that is what we'll end with for this section of genetics. I'll see you over in the next recording.